So now this is the main part which we are going to do today, like for the data collection. As you know, Meghla, today we are going to dive into how scientists gather information or the evidence they need to answer their big questions. Okay, so this process is actually called data collection. They just collect evidence from whatever experiments they are doing. Okay, Meghla. So by the end of the session, you will know the main methods researchers use. Maybe it's a primary or secondary, and how to apply these methods in our research field also, and whatever we are doing in our 3D bioprinting. So Meghla, can you have a look on this? If I just took one tiny biopsy from a patient's tumor, this is a tumor, okay, representing different cells which are in different color. So could I know everything about that cancer behavior and treatment response, and can you answer why or why not?
I think you could find out many things about like the cancer's behavior, but I don't think you could find out everything.
So for example, see, if you can see the needle, it's just pointing out on the blue color. Can you see that?
Oh, it's being sampled like that. I thought that was the whole sample.
See, so okay, I will explain you again. These are different cells in our body. Okay, the red color, the blue color, the purple, the yellow. So for example, I don't know if a patient is having a cancer tumor, and I'm just picking this up for one time, like the blue sample, the blue cells. Do you think that I could get, can I know everything about the cancer's behavior, or can I know whether the patient is having cancer or no? Not by one sample.
Because it's a very small region, you need more than that to identify the cancer and the behaviors of it.
Okay, so have you seen here that I have written different regions represent heterogeneity?
Yes.
So what do you mean by the term heterogeneity?
A heterogeneous solution is where all of them are tightly packed together.
So homogeneous is the same type, right? Like if it was a purple cell, it would be only purple. Heterogeneity means it's like a different kind of cells in your body. So they are representing purple, red, yellow. So these are different type of cells. So I don't know whether the cancerous cells is present in that blue color, purple color, or red color. So if I am picking one sample that is from blue cells, for example, I will not be able to know that the patient is having a cancer or not. Okay, so this comes into the sampling biasness. So a sampling biasness in the cell, it's a systematic error in which certain cells in that population are less likely to be included than others. So your sample is not truly representative. So again, you have to pick a sample from the red color also, and even from the yellow part also. So at least you are picking up 3 samples at a time from different regions of a particular cell. Are you with me, Meghla?
Yes.
Yeah. So did you understand why it becomes important in any scientific experiment, whether it's not about cancer, whether it's about plant also, or you say whether it's about animal research also, you have to always pick up the samples which are in triplicates instead of one sample. So there is a less error, or you can make conclusions which are true at last. Okay, so for example, if the patient is having cancer, say in the yellow region of the cell, so I'm not picking up the sample from that. So what I'm concluding, that the patient is not having cancer because I picked up from the blue one. So for example, in the cancer part, you have to pick up from the the outer layer also, the inner layer also, and the innermost layer also. So then you conclude if the patient is having cancer or not. Okay, so for example, this is called biopsy site biasness. So if in a single tumor sampling only the easily accessible edge I am taking, for example the blue one, so So I'm missing out the aggressive cell, which are also deeper in the core. I don't know if in the blue cells it's a cancerous or non-cancerous. A patient is having all the symptoms of cancer, but in biopsy I'm getting negative when I'm taking a single sample. So at least you have to take samples for thrice from different regions. Any biopsy, it's not about cancer, it can be liver, it can be heart, or any part of the organ. They usually take samples from different regions of a particular organ. I don't want to leave my sample, without concluding. So what is left out, what is not, I would be in a doubt as a scientist. So better to always be in a to make conclusions, you have to be always taking the samples in triplicates. For example, if I am also doing a plant research, for one treatment, I am giving, for example, say potassium as a 10 mg per kg per soil. Okay, so I have to be sure that 10 mg should not be in only one pot. I should be taking 3 pots to average out. For example, in your class, if I say that Meghla's height is say 5'3", 5'4", so can I conclude that the average height of in your classroom is 5'3", 5'4"? No. So what should I do?
You should take more data. You should see more students and see their heights and average it out.
Yeah, then average out, right? Like taking n equals to what? Like at least 60 in your class, or say 10, n equals to 10, then I can average it out at least.
Yes.
Okay. So in science also, every time when you are doing research, always make sure that you are conducting your experiments in triplicates, minimum, like 3 thrice in the same region, in the same type. There can be dependent or independent variables, okay, but you have to make sure that you are conducting your experiment thrice in one go, not at the different time and different region. So for example, if I'm taking a patient sample today, Meghla, from the blue thing, I would be taking the same sample at the same time from the red cells also. Not that you come tomorrow, I will take the samples tomorrow. No, I have to make sure the time, the date of the particular sample is same. Now coming to our what are data collection? There are mainly 2 types of data collection. First is primary data collection and second is secondary data collection. So what do you understand by primary data collection and secondary data collection, Meghla? Do you have any idea about primary data collection or secondary data collection?
Primary data collection is the first round of research where the scientists will perform experiments and they'll take surveys and things like that. And the secondary is when you see the primary data and then you revise it.
Yeah, you are right. Like you have given the whole gist in 2 lines, but you are right. So what is data collection? It's like data collection is kind of gathering information to answer question. Researchers use different types of collecting data. It can be surveys, it can be observation, it can be experiments, it can be interviews, and it can be secondary data also. It also comes in the range of data collection. So you can also ask MCQ or open-ended questions. So this is good for opinion and preferences. Example, I have given here a questionnaire about favorite school subject, or say you want to know which bioink formulations other labs find easiest to print with. You design a short online questionnaire for bioprinting researchers, okay, that ask the above question. So for example, Meghla, you become a scientist in upcoming years, okay, so you don't know where to start from. So you ask your seniors, or you ask your co-workers, or ask your colleagues. You prepare a simple questionnaire that I'm not getting, means that you are not getting the proper data what you thought of. So now you prepare a questionnaire with 2 or 3 questions what you want to learn about, and you ask them, what bioink formulation did you use? Can you just write it down? And what were the problems you faced while preparing this bioink. So you don't repeat the same mistake which they have repeated, right? So you get a clear idea. So other than this, like you can also ask which bio ink base did you use. It can be alginate also, it can be gelatin, methyl acrylate, whatever they used in their lab. So this is the kind of survey we usually do. But in our research kind of thing, the cancer research, your seniors and your guide would be helpful when you are doing such kind of thing, because usually there may be ethical concern if your PI doesn't allow to make a questionnaire, that why you are asking outside your lab, okay, or you have to ask his or her permission for conducting the surveys also around the world. So some people don't want that, the research should not go out from their lab, so they keep the tab on their research also. But basically, while doing surveys, there is no harms while asking the question. So second, coming to the observation part. So what are the observations, Meghla? So it can be in nature, in classroom, or in the lab itself. So have you observed anything up till now, Meghla? Do you want to share your observation part? It can be science, it can be nature, where you observed every day that the bird is making a nest and you observed his parents, or the egg laying process. Any other thing you observed in nature, any flower or bird or butterfly?
The pigeons in my society, I'm not sure if they're ill, but like recently the birds, they've stopped flying away from people. Like
They became wild. They became wild. They were not afraid of the people, right?
Yeah.
So why do you think that happened?
I'm not sure. I've seen people
Yeah.
feeding the pigeons, so that might have something to do with it, but it also might be related to something else.
Nice observation, Meghla. It's like watching and recording what you see as you observed, that some classmates observation, some group observation, and the pigeon observation. So you can also take the photographs of your pigeons, the prints at whatever you have seen or observed, like the pigeons were that at the particular point of time, and the particular point of time they were fed. You can always have this kind of observation. So for example, I've given here the counting the number of birds at a feeder. You usually inspect each patch under the microscope for the uniformity of strength and also the pore size, okay, and take notes, draw or use videos. Okay, nowadays we have this video facilities. Earlier we used to map out on a simple drawing, like you made the mind map, if you remember. So if you have a chance to go through some papers, there are people who have video recorded the cells, how the cancerous cells divide. Have you gone through any of the video? Have you come through?
No, not really.
Okay, so I will send a videotape if possible, if I get one of them, like in a petri dish, how they grow. Have you observed the petri dish anytime, Meghla?
Yes, I've seen petri dishes, and I've seen experiments with petri dishes.
What is the experiments? Now let me be. These are the cause and effect by manipulating one variable and while holding others constant. Okay. So we are testing ideas with control test. So changing one thing and keeping the other same. Then we measure and compare results. So for example, you are testing a plant growth with different light colors. So what is independent and dependent variable here?
The independent, the only thing we're changing is the colors of the light. And the thing that has to remain constant is the care given to the plant and the type of plant we're using. Yes.
Okay, now coming to the interview part. So usually as a STEM researcher or as in science, we have to be also close to the people where, Meghla, we are just doing research in our lab, but the common people don't know how you are researching. So it becomes also important that how you are connecting science and how you are communicating science to the common people. Am I right, Meghla?
Yeah.
Like how you can be simple to connect the dots with the public around you. So what we are doing is the secondary data thing, Meghla. So using data that someone has already collected. It can be books, website, reports and databases. This is for good for our background info and how the trend is going. So for example, using school records for attendance data. Yeah. So now, so have you gone through this Global Cancer Observatory anytime? Yeah. So Meghla, go to what do you call database. I would suggest go to database. I want you to extract the data. If you see the age groups here, I want from 10 years of gap. So 30 to 40, 40 to 50 or 30 to 39 or 40 to 49, 50 to 59, 60 to 69 and 70 to 79 and last May 80 to 90. And if you see the incidence, okay, what do you mean by the incidence rate, Meghla, here?
How often it occurs?
Yeah, so it's like cancer occurring in the population of 1 lakh people. So they count like that, the incidence. So can you see the cancer type here? It's written all cancers. I don't want all cancers. I specifically want breast cancer. Can you click on that? Yeah. So write breast there, you will get it. Yeah. See. Yeah. Okay. So now pick a predefined region. Click there. So say it's Asia. Yeah. Okay. So in sexes, I want females. Acha, are you aware of even male can get breast cancer, Meghla? I only want female from 30 to 40. Sorry, 20 to 30 to 39, 40 to 49, 50 to 59. Wait, can you decrease that manually? So no, go mark it as 30. Okay, I think they have given the specific this one. So for example, if you see the Asia's this thing, can I see India here? So Meghla, I want you to prepare an Excel sheet ranging from whatever they are showing, like what's the range here, 30 to
44.
Yeah, so 30 to 44, then again you start with 45 to whatever the ranges they have given. Okay, so that should be, I think, 30 to 44, then 44 to 54.
Yeah, I believe.
Or 43. Sorry, 53. Extract the data. So I will be giving you the pura how to do it. See, it's a predefined selection. So be careful while resetting everything. And then just download the Excel file for Asia, and then you extract the India part. So first you collect the raw data from here. Okay. So age group should be
30 to 44 and then so
then up till 80. So for the literature analysis part, you have to include 2 more research papers, not review papers, because we are going to count frequency of the cancer cell type used in n equals to 10. So how did you find today?
I found it interesting.
Yeah.
I like collecting data. It's nice reading all of it.
I think it can be useful when if you want to do research in your school also now for science day, or you want to display your project also. You are always free to ask me whatever you want to do in your upcoming projects also. Yeah? Okay, Meghla. Chalo. Have a nice day.
Thank you, ma'am. Bye, ma'am.
Bye.